Okay, I want to spend a little bit more time looking at methods related to proteins and their structure and their detection and their their like three-dimensional folding as well. We've talked a little bit about these methods, but I want to kind of break it down for you guys so you understand where these methods fit in if we're talking about looking at a protein. So, a lot of the methods we discussed, and we discussed these in the previous chapter and have mentioned them here as well, are methods to purify proteins. So, again, all these methods are doing are helping you separate out proteins of interest. You have some mixture of proteins, you're doing an experiment, and you have um, some biofluids, or you have a cell that has a bunch of different proteins in it, but you only want to look at maybe one of those proteins. You're studying that one protein. Or, for example, you just created a particular protein. You synthesize some insulin in a cell, but you need to separate the insulin out away from all the other proteins that are in that cell. You would use a protein purification technique. So, again, these are techniques like ion exchange chromatography, size exclusion chromatography, affinity chromatography. Even gel electrophoresis can be used for purification because as you run through the gel, you, of course, will get your separate bands as the protein moves through the gel. So, you would be able to separate your protein this way. You can also separate protein using things like solubilization. So this is key in things like using the PI to um, make the protein become insoluble. Remember, at the PI, the protein's charge is 0. And at a charge of 0, proteins are insoluble in an aqueous environment in water. They will not stay dissolved, they will not stay soluble, and they will precipitate out. So you can use, for example, something like protein solubility by adjusting pH to essentially precipitate out a protein, and that will help separate it from other proteins that have different PIs. So again, we've talked kind of about these techniques a little bit, and these are all in the realm of purification or just the ability to separate out a protein. What if you want to detect if there are proteins there? Well, there are two kind of main classifications of detection. There's very non-specific methods. So these methods are just going to tell you you have this much protein. It's not going to tell you what protein it is. It's not going to tell you you have this much of protein A versus protein B. It'll just tell you that you have 100 grams of protein in general. So these are very non-specific methods. A lot of methods are going to use absorbance. So you can just put a sample of proteins into a spectrometer and read the absorbance. And remember, we can detect a lot of proteins because they have these aromatic amino acids. Tryptophan, for example, tyrosine, phenylalanine, all of these are in that aromatic group and they will show up on an absorbance spectrometer. Again, you can't tell how much of a specific protein there is, but you can just tell like a general concentration level. Other assays that are available to help you tell concentration of total protein are things like a Bradford protein assay, Burette test, this BCA assay, and the Lowry protein assay. All of these assays are very similar. They use a color determination test. So they take some dye that will react in the presence of just protein. Again, not a specific protein, any protein. They're not picky. And so you just measure the kind of color change, again, using absorbance to detect generally how much protein there is. We'll actually be doing a sample of this in the lab. We'll be using one of these color dye tests to detect how much protein there is in a sample. So again, these are types of detection methods, and these are non-specific methods. Now, there are specific methods of detecting proteins. This is if you really want to know how much of one particular protein that you have is in a sample. So things like spectroscopy using HPLC, that's high performance liquid chromatography. This can single out separate proteins, and essentially it's separating and then doing detection all in one run. And also mass spectrometry. We tend to kind of combine these techniques together. We'll run a mixture of proteins through a column, we'll separate out them into chunks, and then we'll put it into a mass spectrometer and use the mass to determine which protein is which. And that way we can say, oh, here's my protein A, and this is how much of it I have. Now, one of the other main types of methods to detect proteins, which I really want to spend some time on, is antibody methods. There are a couple types of antibody methods, and generally they are going to be using an antibody that's built for the protein you want to look at to detect that protein. Some of these examples are ELISAs. You'll hear about enzyme-linked immunoabsorbent assays, ELISA assays, and then hopefully in our lab, we'll be using an ELISA assay to look for a protein on COVID-19. So this will be doing an antibody test to look for a protein on COVID-19, which will tell you how much of it there is in a sample. Other examples include Western blots, which are very, very, very similar to your gel electrophoresis, except instead of those bands being labeled just based on how far they moved down a gel, they will be highlighted, or they will actually be colored if it is the protein that you want. And we'll be talking more about Western blots. Western blots is a very key technique. Again, the same principle, you use an antibody to look for your protein. Also, things like staining, immunostaining. We can stain a whole piece of tissue or a whole cell, just staining the area where that protein is on the tissue or on the cell. Lets us get a whole picture of like where are proteins expressed in a tissue or in an organ or in a cell. Where do they kind of live? That helps us detect proteins when we're asking questions like as such. So something like immunofluorescence, you see a lot in cell biology. Again, this is where we're using an antibody to stain for a protein in a cell, and that antibody just happens to have some fluorescent tag on it. And you get to see pictures like this. So here are a bunch of cells. The reason that we can kind of see their structure is because an antibody with a green tag on it was used to label proteins in their membrane. And then another antibody with a red tag on it was used to label proteins in their nucleus. And so you kind of can see exactly where those proteins are expressed via their color. So this is the general layout of how these antibody-dependent methods work. Again, you have some protein that you're interested in, right? And we want to know how much of this protein is there. And what we will do is attach a primary antibody. This is a specific antibody for that protein. Um, these antibodies are usually custom made to detect that protein specifically. So you'll add it, and then you will then add a secondary antibody. This secondary antibody is custom built to bind the first antibody. So it will only bind that first antibody. So you put your primary antibody in, it will bind only the protein that you're looking for. It won't bind any other proteins. You then put in your secondary antibody, it binds just your primary antibody. And so now you kind of have this stacked antibody. Secondary antibodies usually are made with some type of fluorescence tag attached to them. And when the secondary antibody finds its primary antibody target, it will fluoresce. So in this way, we call this kind of sandwiching or stacking of antibodies to look for a protein. We can determine how much of a protein is present by using the fluorescence or the color that is shown when the antibody binds. Okay, so the last type of question that you can potentially ask, you've already know how we separate proteins. We already talked about how we can detect the amount of protein, whether it be the bulk amount or to detect a specific protein. But what if you want to know the actual structure of the protein? You guys have been looking at structures of proteins. We have been using the PDB, that protein data bank, and it has tons of structures of proteins. But how did we actually get these structures? Well, there's a variety of methods, and the two that are the most used and most popular are X-ray crystallography and protein NMR. These two are kind of the main bread and butter for determining protein structures. One that is gaining more recognition and being used more often is this cryo-electron microscopy. Small angle X-ray scattering and circular dichroism are still used, but to a much lesser extent now. So let's talk about these methods because you guys are going to be seeing these structures a lot. So it's important to understand where they're coming from. Again, one of the main staple methods to determine a structure is called X-ray crystallography, which kind of does exactly what it sounds like. It takes a crystal and it uses an X-ray to examine the crystal structure. In order to get a protein to be measured using X-ray crystallography, you need to first, of course, purify the protein, and then you need to crystallize the protein. This is probably the hardest part because not all proteins are easy to crystallize or easy to separate out and stay crystallized by themselves. If you do get a crystal of a protein, congratulations. You can then use the X-ray to collect diffraction data and then kind of calculate the structure and electron density of that particular protein, which will then essentially be used to make a map of its structure. Some pros to using X-ray crystallography is that there are no size limits of the protein. You can look at the crystal structure of anything small to large using this technique. It's also very well established, so we have a lot of methods that are kind of already built in, and we very well understand how X-ray crystallography data looks and being able to interpret structure from it. Some downfalls, some cons on using this type of method is that it's very difficult when you have a protein that's embedded in the cell membrane. Some proteins live in cell membranes and they're very hard to separate out of the membrane while still retaining their structure. So they don't crystallize well and they just are very hard to actually kind of get a grasp on their whole structure using this method. Another con, it can be said as a con, although it's usually not that big of a deal, is that you can't actually see hydrogens using X-ray crystallography. X-ray crystallography kind of gives you points along the structure, and it's based on their electron density. So the more electrons an atom has, the more dense it looks on this type of method. Hydrogens, of course, are very small. They only have one electron, and they basically show up as nothing on these structures. Usually that's not a big deal. We kind of just assume based on all the other elements where the hydrogens are. But if you really need to know where a hydrogen is moving or how a hydrogen is involved in some type of structure, this would be a con. Now, the other main type of structural method that is used is NMR. NMR, again, to be able to run an NMR, you have to purify the protein and then you have to dissolve the protein into some type of liquid, which is usually quite easy. So, getting a crystal is much harder to do crystal X-ray diffraction, but just dissolving a protein in some liquid is very easy. Once you do that, you collect the NMR structure or the data, and then you can assign the NMR signals to find the structure. Now, because these structures are in liquid, you can imagine a crystal does not move very much, it doesn't move at all. When you see an X-ray crystallography structure in the data bank, it'll look nice and clean like this. And that's because when we measured the protein, it was frozen in a crystal. When you measure a protein in NMR, it's in a liquid, and so it's kind of moving. So NMR structures will look like as shown. They kind of have this wiggly looking structure, and that's because we're kind of looking at the protein in maybe 10 or 15 different varieties as it's moving in liquid. So one pro, of course, of NMR is that you don't have to crystallize the protein, which again, sometimes proteins cannot crystallize or they're very, very hard to crystallize. And you can also see hydrogens in NMR. NMR is actually excellent at picking up hydrogens because it's using protons to and their magnetism to look at the structure. Some cons in NMR is that some proteins can be insoluble. They won't stay in solution very well. And in this type of experiment, of course, you have to keep it in a solution to run it. NMR also works best when you have only small proteins because assigning the signal to a protein with NMR versus a crystal structure with X-ray is a much longer task to do. So having a very, very, very large protein to look at with NMR and figuring out its structure is a task that takes potentially years versus small structures, which would only take maybe months to determine its structure.